We'll go ahead and get started. I want to say good afternoon and good evening to everybody. My name is Theo, and I'm the Director of Scientific Affairs here at Ovarian Cancer Research Fund. Thank you all for joining us as we continue our webinar series for 14. And tonight's topic is about some exciting new findings in small cell carcinoma of the ovary. Our entire webinar series has been supported by Genentech, and I want to thank them for their generosity in making it possible. So it's the largest and oldest charity in the U.S. funding ovarian cancer research. Our mission is to fund scientific research that leads to more effective identification, treatment, and ultimately a cure for ovarian cancer. And since we made our first grant in 1998, we awarded over $57 million in research grants. Before we get started, I have just a few housekeeping notes to go through. All listeners will be in a listen-only mode throughout the presentation. Some time for questions after the presentation. So if you think of a question as you're listening to Dr. Levine's presentation, please just type it into the Q&A box that you can see on the right-hand side of your screen. Nobody will be able to see the question that you ask, and you won't be able to see anyone else's questions, but I will see them all and select some to ask Dr. Levine at the end of the presentation. So there's no until the presentation is over, you can just type questions as we go along and as you think of them. I'm very pleased to have Dr. Doug Levine with us tonight as our speaker. Dr. Levine is the head of the Gynecologic Research Laboratory at Memorial Sloan Kettering Cancer Center here in New York. He's also an associate attending surgeon. He's a gynecologist who specializes in the surgical treatment of women with ovarian or uterine cancers. And in the lab, he focuses his research on genomics of ovarian and endometrial cancers. He's a very researcher, and his research has been supported by all the major funding agencies, including National Cancer Institute, Department of Defense, Stand Up to Cancer, Industry Foundation, and, of course, CRF. He's author of peer-reviewed research papers, articles, book chapters, and founding editor of the Atlas of Procedure in Gynecologic Oncology. He served many editorial and professional boards, including co-creating the Cancer Genome Atlas's Ovarian and Endometrial Working Groups, and he serves on the Scientific Advisory Committee, where he generally gives many hours of his time to help us with our work, and we're very grateful for that. So, Dr. Levine, I will now pass it to you, and you can get started.
And please feel free to interrupt at some point. My sound drops off or anything like that because you know who you're talking to. And many of you may not know who you're listening to. I will try to flip on my here. Whoops. Work out. There we go.
I can see you.
I love it. That you can see me. Turn off my again so as to not be a distractor, but good evening, everyone. Sarah and the OCRF for letting us do this. I actually approached her because so many people are so generous in the course of the study that we're going to discuss tonight and some of the other studies. It's very reasonable to make sure we shared the results with everyone and had people opportunity to ask questions because there's some very interesting and important findings, I think, from the study. So let me try to get started and share my deck. I just have to figure out which button to click to do that. Over here?
Yeah, if you go over to the Quick Start menu, there should be, which you can click
yes.
to on the top
great.
left.
Okay, great, perfect.
There you go.
Okay, in 1 second. There. And you can, right here it comes. Okay.
Looks great.
So we're going to talk about SMARCA4 mutations in small cell carcinoma of the ovary, work that I've done with people at Memorial Sloan Kettering Cancer Center. You may notice on the bottom that we removed the hyphen from our name a few weeks ago.
Oh.
And logo. So let me see how this looks. Let's see. All right, so what I'll do tonight, probably in a half hour, I would imagine, is review the scientific data from these recent discoveries. And I'll take 2/3 of the time, maybe half the time, discuss the implications for therapy in treating this disorder and implications for genetic testing for patients and family members, and answer questions. So many people on the phone probably know that this is an aggressive ovarian cancer, which is why we really need to improve our knowledge about this disease. If you don't know, it affects young women, very young women, unfortunately. After diagnosis is about 23 years old, and advanced stage disease often recur within a year or so, despite our best treatments, and do not, of course. And familial cases have been reported in literature, meaning multiple family members have the same very unusual tumor. A lot of pathologic features. Robert Scully, who just passed away last year, used when he first this disease, and can be a very difficult diagnosis for pathologists to make because it's rare, and also because it can be very similar to other types of cancers. Whether mutations that exist in this disease, whether they occur more than they're recurrent, we can in the future use that information to improve other management of this disease. And the scientific story is that Liz Oppo, who runs the Katie Oppo Research Fund, came to us and someone needs to work on this disease. And we're very happy that she came to us, and we're very happy that we started working on this because it's been very worthwhile. So that's actually how we actually got involved in studying this disease. It's one of probably many examples now where actually patients and patient advocates are critical partners in research and for getting scientists like me to work that I might not otherwise have worked on. And now make this an important part of our laboratory efforts go forward. Cases and study them. And a lot is genomic science, and so we sequence things, we identify mutations, and we look at gene expression, we look at protein expression, do all sorts of genomics. And so we scoured the hundreds of thousands of patients at Memorial Sloan Kettering, and we found a total of 3 patients who had some of the ovary hyperthymic type. And just again, as a point of background, of all the small cell carcinomas of the ovary, about 2/3 are hyperthymic type, and about 2/3 of those actually have high levels. The type of small cell carcinoma of the ovary called neuroendocrine type, which has similarities to lung cancer. Small cell carcinoma of the ovary hypercalcemic type is not that, and talk is all about the more common subtype. I mentioned small cell carcinoma of the ovary hypercalcemic type. 3 cases. We knew that wouldn't be enough, but we started 3 cases. We knew from the tumor what's called germline DNA. So to bring up to speed, inside the tumor itself can develop various mutations. Those are called somatic mutations because they occur in the tumor, and for the reason the tumor can be a cancer. Every region in the rest of your body, and the DNA in the rest of your body is called germline DNA. Meaning, no type of DNA is present in all of your whole body, whether it's a mouth cell or an intestinal cell or a cell. The germline DNA is what you inherit from your parents, and it's present in all parts of your body. And in some situations, germline DNA and DNA will work together. But I'll be using the term germline DNA, and that's basically the DNA you inherit, and it's in every cell of your body. So types of specimens to compare what is unique to the tumor, and what is unique to the tumor is just the fact that makes you tall or short, fat or thin, have dark hair, and things are part of every cell of your body. So we knew sent a call, and again, the Marion Peterson Cell Ovarian Foundation, who I believe cannot be with us on the call today, was generous to spread the word through her network. And 9 patients over the course of 6 months or so allowed us to use their tumors, and so now we had a total of 12 cases. And we started with the first 3. We do sequencing where we can look on DNA many times. So we find all patients that may be low frequency and that may have been overlooked by technologies that we didn't have available to us just a few years ago. One of the colleagues who does this type of sequencing called me a few months later, and he said bad news. They didn't look at the data for a few months. The good news, 3 patients have the exact same mutation, and this is unusual. It's not only that all 3 of these patients have the same gene that's mutated, but a few other mutations in any of the other 300 genes that we looked at, and this does not happen very often. So we quit the next 9 cases. It turns out it wasn't such a big deal that he waited so long because it took us that long to get cases, and we sequenced the additional 9 cases. In fact, all of them had a mutation in a gene called SMARCA4. On the side, you can see the 12 patients who were sequenced, and they are various types of mutations in the same gene. And genes that we sequenced, and the point is not to have everyone read every single gene, but we looked at all the major genes that are thought to cause cancer, and of all, a few of them were mutated, except for the gene here, which is SMARCA4. And I'll talk about this gene, SMARCB1, a little bit later. So we find different types of mutations. You can see them here. Splice mutations are important for turning DNA into RNA. Home deletions are where large portions of the gene is missing. Mutations are where a mutation causes the stop turn into a protein prematurely. And one type of mutation that we didn't find is called a missense mutation. You can see it over here, different gene. And missense mutations are not always so important. Some may be important in some genes. They may not be so important. But all the mutations that we identified are what's called inactivating mutations, meaning they're likely to affect the way a protein functions. Showing you that of all these cases that were each sequenced,
Your audio just went out. Hear me, but if you
no?
Working.
It looks like
Yep.
I don't know if you can hear.
Best to dial in. Everybody, we get Doug's audio sorted out. Perfectly.
Okay, I'm going to hold on one second. Microphone said that it was working, so maybe it was an internet issue or something.
Could be.
You can hear me now?
I couldn't hear you for 30 seconds there. But
okay. Can you see the slides?
Yeah, perfect.
Okay, because I was anyway, we're here, so I'll just use this. I don't know what you heard, but I think I was in the middle of saying that there were very few other genes that were mutated, and usually we would expect a lot of genes to be mutated. let me know if the sound causes any other trouble. So just moving on. Again, sorry about that, everybody. Modern technology. What we next generation sequencing, and you can see on the left side here, this is a normal sample for one patient, and this is a tumor. And so normally at this location, there would be a cytosine, and this is turned into thymine in the tumor, and that's a mutation. It's present at a very high frequency here on the bottom, and this graph really represents about 400 different reads of a population of DNA. And so this is our screening test, how we can pick up mutations very quickly and easily. And then on the right side, you see something called Sanger sequencing, which is sort of old-fashioned sequencing. And every single mutation we validate to make sure it's not an artifact. And again, you can see at this location, there's supposed to be a D, and it turns into an A, and that's because we're going on the reverse here. And you can see a heterozygous mutation, meaning that sequence left, and there's a mutation on top. And so every case basically gets validated like this. We also get the protein, because the protein is really what causes the function inside of a cell. And this is one case, not necessarily the same case, but normally you'd expect all these cells that my mouse is going over here, you'd expect them to be very dark. Like on the bottom, this is a regular ovary cancer, typical high-grade serous ovarian cancer that has intact SMARCA4. You can see it's very dark, and here it's very light, and that means the protein is lost. I guess this slide, here it is. This slide basically shows you that in this one case, we found a mutation in the normal tissue. So that's called germline mutation, and you can see in the normal tissue, which may have been a blood sample or some other type of normal tissue that did not cancer, it has mutation and the regular part of the gene that's not mutated. And in the tumor, you lose what's called the wild allele or the non-mutated allele, and all you see here is the mutation, and that also results in a loss of protein. So this is only one case we had where we found the mutation in a normal blood sample or a sample of some other type of normal tissue, and we'll come back to that in a few minutes. We also see, as I mentioned, large parts of the gene can be deleted. There's 2 exons that are deleted. For those of you who have familiarity with genetics, you can see that this puts 2 different exons together, which should not happen normally. And finally, I'll skip this slide. What I just want to show you here is this is called a Western blot in the middle, and this is another way that we measure protein. So in our first 2 cases, we had frozen tissue available where we could do these types of protein studies, and just like this cell line, which is actually a lung cancer cell line, these proteins are completely absent, again suggesting the importance of this mutation in causing the tumor. List of all the mutations that we found, and then to let you know what this gene does, which is the common question. This is part of a complex called a SWE/SNF chromatin remodeling complex. So chromatin regulates whether the DNA is very compact and is protected versus whether the DNA opens up and the RNA machinery can come in and turn the DNA, which is the instructions, into protein, and it's the RNA that helps turn DNA into protein. But the has to open up for this to happen. And these chromatin remodeling genes, of which there's a number, and you can see them down here in this little part of the figure, there's multiple subunits. And so within this, one of the genes is SMARCA4, which is an important gene, just like the other ones are important protein. And if that's mutated, then the cancer cannot regulate the process of making proteins properly. And that's what this gene does and other genes do in this complex, which again I'll mention gene ARID1A mentioned later on, as well as SNP5, which is also SMARCB1. The next thing we do is we say, well, is this very uncommon? So we now close to 5,000 tumors that have been sequenced by a nationwide study, and so is across how often is this gene mutated, and you can see that usually about 5 or 10% or less will we find mutations in this gene, and in our study of only 12 patients, we found it 100% of the time, which is very unusual, and these are some of the other types of cancers where this gene can be mutated. and many of these tumors have the blue bars here, which are called missense mutations. And so it's not entirely clear whether these mutations really result in loss of the function of the protein, whereas all of these types of mutations are inactivating mutations. And so we did some laboratory studies that we're working on. I don't know how much detail I need to go into this, but basically we can manipulate this gene, and we can get rid of this gene over here and make the cells grow faster. We can take this cell line that I showed before that doesn't have the gene, we can reintroduce it and make the cells grow slower. So this is, again, proof of principle that this is a very important gene for growth of tumors. That came out probably 3 weeks ago or 4 weeks ago now, maybe. This is our paper. We found mutations in 100% of 12 tumors. We're sponsored by many foundations and funding agencies, and the Katie Apple Research Fund was our initial funder here, and these are some of our other funders that support our work. At the same time that this paper came out, 2 other papers came out, and we are all sort of friends in this relatively small research community, and so we actually were in contact with each other a little before the papers came out because we knew about each other's work. This is a very excellent paper from Will Fuchs, who's at McGill in Canada. He is a cancer geneticist who starts by studying rare tumors that occur in families, and so he's one of the people who's previously reported the familial cases, and there's a very small chance that he will join us later at the end of the call. He had a conflict. He was going to come join us today. So I guess if he's on, I would just suggest him to send a message to Sarah, and you can make him a presenter later on, and we can try to remind him of that if he joins in. Otherwise, he'd be able to hop on here. So he started with a few families, and he found that every affected member of the family actually had a SMARCA4, and then he collected a number of other cases, actually 32 cases altogether, and 30 of those 32 cases also had these mutations. The other cases were some from families, some were non-familial, and some it was uncertain if they were familial or not. He found that about 50% of his cases mutations in the gene, which is quite important, and leads to certain consequences. So 50%. Now that may be the right number, or that could be a biased number because, again, he studies families that vary diseases, including rare diseases, and so families that have these diseases may be going to him, which might be why he's finding a higher rate. And he and I discussed that recently, and we don't know right now what the exact frequency is of hereditary case versus having a sporadic case or a non-hereditary case, and that's something that we certainly need to figure out. And now that we know the gene that causes the cancer, we're better suited to figure that out in the next little while. A third published by David Huntsman from British Cancer Agency in Vancouver, and Jeff Trent at TGen out in Arizona. They all had 12 cases. They found 9 of them had these mutations, but they have the normal tissue to compare to on all of their cases, and it's not entirely clear which patients had the tissue and not, but they also basically found the same findings of these other reports. So we have 3 reports all the same findings, and basically we're all confirming each other's results, which is kind of unusual to happen, at the same time. So we can conclude that this is real, and we can conclude that this is the cause of this disease. These other papers did also supported the fact that there's very few other mutations in these tumors. And so unlike the lung cancer or the bladder cancer where there are some of these types of mutations, they occur in the background of many mutations, whereas these tumors have very few genes that are mutated besides SMARCA4. We conclude our showed 100% mutations, indicating this is the driver mutation for this disease. Now, SMARCB1 is mutated in almost 100% of a related tumor called pediatric malignant rhabdoid tumors, which is also a very aggressive type of tumor. They look very similar under the microscope, and so this is a variant of a rhabdoid tumor, which is a variant of a sarcoma. That's an important finding also.
I think I'll mention that in just another minute as well. Um, and, a substantial or some fraction of these cases derive from germline mutations, meaning inherited mutations. It's a rare tumor, and we don't exactly know what portion of these cases may be from germline or inherited mutations. And of course, we need to identify therapeutic targets, and we're working on that in our laboratory. And I know the British Columbia folks are working on it as well. And so we hope to make progress in that area. To the future directions, where do we go from here? Obviously, this is a very important finding, but what do we do with it? How can the finding impact our research and ultimately lead to therapy for patients and women who actually have small cell carcinoma of the ovary, hypercalcemic type? So we're looking into what are the mechanisms to target mutation, what are the consequences of the mutation. We probably can target this gene directly, but the question is, what does this gene do? What other events does it cause that makes the tumor grow? And then can we interfere with the function of the gene to reduce the tumors, make them less and shrink, and ultimately, obviously, we'd like to make them go away and never come back. But usually the first step along the way is to find an effective therapy that reliably treats these tumors, because we know what drives the tumors. This, unfortunately, is a long process, so as I'll mention, I think on the next slide or so, as of today, this finding does not help us treat patients. This finding helps us lead to the work that will ultimately allow us to figure out how to treat patients. We're taking two approaches to this question. One is called the candidate approach. So we test what we think we know. We think we know what this complex does. We think we know what happens when this gene gets mutated, and so we're going to try to test our ideas and see if we're right, and unfortunately we're usually not right, but we do try a candidate approach. The second approach we take is an unbiased approach. We can look across the entire genome and see what else happens when you mutate this gene. We screen lots of drug compounds through high throughput screening, which is somewhat of a lengthy and extremely expensive process, but we can screen lots of drugs and figure out which drugs actually work the best when we introduce this mutation into a cell line or an animal ultimately. And studies are limited by financial and scientific resources. As I mentioned, some of these studies are quite expensive, but the scientific resources are equally problematic in that we only have one cell line from this type of tumor. The cell line happens to be called BIN67. It has been sequenced, and it does have a SMARCA4 mutation, so it all makes sense. Like more than one cell line, so we can sort of confirm our findings or study a population of different types of cell lines. But unfortunately, since the tumor is so rare and cell lines are hard to develop, what you really need to do to develop cell lines, which we can do nowadays, is to get a fresh tumor that's immediately removed from a person and immediately start to grow it. Since many people have initial surgery all over the country and not often at major cancer centers where these cell lines can be made, it's very hard to make a cell line. This cell was actually made by accident when a number of investigators from I want to say the late 70s, but it could have been the 80s, were trying to actually make sarcoma cell lines, and they made this cell line by accident. We're doing I mentioned the unfortunate fact that we don't have a therapeutic approach today, and that it's going to be difficult. But the news is that there are other genes in this complex that are frequently mutated, and lots of people are working on those genes as well. So one of the genes I mentioned, called ARID1A, is mutated very frequently in clear cell ovarian cancer. And again, malignant rhabdoid tumor is a close relative of this disease, and people are working on SMARCB1 because that's been known for a number of years. So these research areas will overlap and will help us to do our research into small cell carcinoma of the ovary. Personally, I'm interested in looking at people who have had this disease and have present at an advanced stage and are doing very well and are cured or surviving for many, many years, because that's uncommon. So I'd like to study those tumors and try to learn what allows those tumors to do well and see how we can apply that to treat the tumors from patients that don't well, and to learn more about how we can target this cancer. I've spent the last few minutes talking about clinical implications. So what implications might this have on patients or family members as far as a genetic testing perspective? So here's what's called a pedigree. It's a 3-generation pedigree. I think you can see my cursor, and Sarah, correct me if you can't see me or hear me. This is great. This is the so males are squares and females are circles for whatever reason. And these parents had 3 children, and I believe 2 of them were affected with the disease. And this person married and had another child who also developed the disease. Now, people in black who have the cancer, they all have a mutation. So the mutation came from either the mother or the father. The mother who does not have a mutation because that person's been sequenced. This is actually a figure from Will Fuchs's paper that was published at the same time in Nature Genetics. And so I just show this figure to indicate there are families where you'll see multiple people affected and they all have the same mutation. So the question is, this person over here also is a mutation, and I believe, I could be mistaken, but I think this person or someone like this person actually had their ovaries removed to prevent this disease. And may or may not make sense, and we probably won't talk about that too much today, but you could imagine if you had 2 sisters who had the cancer and you have the exact same gene mutation and don't want to get this cancer, that might be a reasonable thing to do and gets into quite a complicated situation. So we can assume that the mutation came from the father. The mother did not carry the mutation. Now, of course, the father cannot get this disease because he doesn't have ovaries, and so he's the carrier that passed on this mutation. And what to do about this child who also seems to carry the mutation? That's obviously a very personal and important question to discuss. And so what we know is, will this person or this person actually get cancer? Now, this family has a lot of cancer, so you might imagine these 2 people are at pretty good risk of cancer. But let's say this family only had 1 cancer, and you had a sister who carried a mutation. Is she very likely to get cancer, or is she not likely to get cancer? And that's called penetrance. It's the proportion of individuals carrying a mutation who also get the disease. If we think about BRCA1, which is much more common and well known, that gene that can cause breast and ovarian cancer. If you carry a BRCA1 mutation, your approximate risk of ovary cancer over the course of your lifetime is 40%, and that leads us to recommend removing of the ovaries, at a certain time in people's life. That's the penetrance. It's 40%. So 60% of the patients will not get ovary cancer, but ovary cancer is also very aggressive, and so therefore we recommend removing tubes and ovaries from people with BRCA1 mutations when they're in their 40s generally, or 40. The penetrance is unknown because we just discovered the mutation, and we don't have enough patients to determine this. But what we really need that would be very interesting is to find an old, unaffected female. So if you go back here, if this person was a woman and mutation and was, let's say, 80 years old and never got the cancer, that would be called incomplete penetrance. So this would be a person who could get the cancer mutation and did not. Now, this is, of course, a man, so that can't happen. So now, me, my colleagues, and the other investigators, we are not aware of an old person with mutation who does not have disease. Now, very rare. We just discovered the gene, and so these people could exist. But it would be interesting to test older women in families to see if they have the mutation and they did not get the cancer. And that's currently unknown and unclear, and that will be an example of what's called incomplete penetrance. So we need more studies to do this. The true penetrance, and we don't know really the true frequency of germline mutations, but we'll probably be able to figure that out in the next few years. I think it's reasonable to have genetic testing. There's various ways to do this, but if you were a patient with this disease, we can very easily look at this one gene and figure out whether it's just in your tumor or if it's in every cell of your body. It could be inherited, and if that's the case, then other family members can be tested, and if a patient has a mutation and a family member does not, then they at risk for the cancer or not at an increased risk for the cancer. If a patient has the mutation and a family member does, that's a little bit unclear, but the family member might be at increased risk for this mutation. We certainly can figure out which cases are related to germline mutations and which cases are related to somatic mutations, meaning they just developed within the tumor. What is tricky? Unfortunately, this cancer does affect younger people, so there's lots of obvious concerns, and we don't have all the answers. We take the approach that getting the knowledge is the first step, and we have to individualize recommendations. And so all of this type of genetic testing should be done under the direction of a genetic counselor or a cancer geneticist. They will understand exactly what all this means, and then decisions should be made in consultation with also a GYN oncologist, who's someone who actually can perform various surgical procedures that may be necessary. The best outcome, of course, is if one good outcome is if the cancer is not hereditary, then that sort of puts that issue at rest. If the carrier and the family member is not, that also puts the issue at rest. But if people are going to start being tested and family members are carriers, that really is embarking on an unknown territory that we have to encounter together as medical professionals and patients and family members and kind of work together to figure out what's going to be the best thing to do. So just to conclude, this I think is my very last slide. Genetic testing should be considered for patients or family members. We test family members directly for the one gene. Better to test the patient if possible. Qualified genetic counselors need to coordinate the testing for this gene. And for both, by patients, families, and researchers may be required because this is completely new. And if you were a patient or a family member who walked into your genetic testing and said, I'd like to be tested for SMARCA4, they would probably look at you kind of funny. But if then you explain to them what's going on, it would make perfect sense. And I'll end by saying that a new paper that just came online, again by Will Fuchs, who's doing great work in this area as well, on the genocide, he's suggesting that this tumor is so close to the malignant rhabdoid tumor that occurs in pediatric population that tumor should be renamed malignant rhabdoid tumor of the ovary, and I think that's a reasonable suggestion. Just saw the paper. I think it came out a few days ago. I have to read it, but that may be something that will happen in the future. I do this work with my laboratory. I'm a G1 oncologist. I treat women with ovarian and endometrial cancer. I also perform research with these lovely people here who do a lot of the work. This is Jenny Mueller. This is me. And this is Petar, and the two of them were the driving people on the project. Fanny Dow helps with a lot of our studies, as does Narciso and Faina and everybody, but these people work together. We have a great team. Narciso pulls tissues and cuts blocks. Faina extracts DNA. Fanny coordinates all the research activities, and then together we do all the sorts of laboratory experiments, and you can see here all the people who fund work. And with that, I'm going to stop and minimize and try to stop sharing my screen here. And there we go. Stop sharing. I think I stopped sharing, and turn it back to you for the moment.
Okay, great. So much, Dr. Levine. It was a great presentation, very comprehensive. There are a few questions that came in just to kind of clarify in terms of genetic testing. There are a bunch of questions about this. One driver who was diagnosed in 2005 is asking if should her parents be tested for the gene mutation?
That would be very interesting. So first we could test her just to make sure. I mean, there may be a few cases out there that don't have mutation. So why might someone not have this mutation? Maybe just a really weird tumor. We've only studied collectively less than 100 of these in our 3 papers. Or wrong diagnosis. So this is not the easiest diagnosis to make, and I could imagine 10 years ago, 15 years ago, maybe someone got the wrong label. So the first thing would be to test this person who may or may not have been in one of these studies, but someone would easily test. So there's research and clinical. So I would be interested in looking at the parents to see if they're a carrier, particularly the mom or the dad. But the patient tested to remember the difference between germline and somatic. So the patient will be tested to see if her case is sporadic or somatic, or inherited. If her case is not inherited, there's no reason to test the parents. If her case carries a germline mutation, then testing the parents would be very worthwhile and from a research perspective. And I think just for everyone on the phone, on the call or web, when registering, we asked if we could contact you in the future, and this is exactly the reason why we would do this. From a point of view, with some, with some heads, I'll say that we, be contacted about various research issues. We can't really give out clinical information directly, informal. We're not allowed to do that. Obviously, on the call, we're going to answer general questions. But from a research point of view, we'd be very happy to be contacted to help further the research in this area.
Okay, on the phone, just sort of coming in, I would like to be contacted. You can also, depending on how you answered the form, that will be helpful info for Dr. Levine, but you can also just contact me if you're interested, and then I can pass your contact info along to Dr. Levine as well, because we have some other asking about, connected and this sort of thing. One other question. So someone wants to clarify. So a son can pass this on as well?
Yes, so any gene, everyone has the genes. Anyone can pass them on, just like BRCA mutations, but you may or may not get the cancer. So for BRCA1 is a great example. I could be a carrier of a BRCA1 mutation, but I'm not going to get ovarian cancer. But I could easily pass it on to my daughters, who could then get it if I had daughters. And so we like to test everyone to know who might be at risk. But yes, men can pass on any genes. And I think, can you see my slides again, Sarah?
Yes.
With some hesitation, this is our lab email. It's, sorry about that. It's GYN Research Lab, spelled like this, at mskcc.org. So for research-related questions, for people who would like to help us with our research studies in any way, feel free to contact us. It's a small community. We all help we can get so we can work together. So do feel free to email us. Please don't ask us clinical questions. We can refer you to people in your area who can help with that. Happy to discuss any research activities with people.
Thank you. Okay, hold on. We have some other questions here. Let's see. Here's a question. While treatment may not be available for, say, 3 to 4 years, as you said, what is possible testing to determine if a survivor has a recurrence, i.e. in place of a PET scan?
Yeah, that's a good question. That also applies to many types of cancers where we know what mutations exist. And so we are working in a research setting on things like taking a blood sample and trying to find what's called circulating DNA and those sorts of things. That would have to be developed, which we probably wouldn't do in this disease because it's quite rare and it's not even, really used clinically for any cancer right now. So again, PET scans and CT scans are really going to be the best surveillance method. So I don't think we would do from PET scans and CT scans. Some tumor markers, again, like CA125, it may or may not be elevated in some patients. Anything that's elevated at the time of diagnosis is a good tumor marker to use for recurrence.
Okay. New question. One of my daughters passed away from this disease. Would it be beneficial if my other 2 daughters had their ovaries removed, just to be on the safe side?
So it gets really tricky. So the simple question is that, and this is a little more complicated. I'm not 100% sure this can be done, but I think it probably can. So, and I'm very sorry to hear about the person whose daughter passed away, by the way. If specimen from the tumor was available, that tumor could theoretically be tested. There are companies who will do testing. These companies will do testing on what's called archival tissue that's just being stored somewhere. That tissue probably exists. We can certainly do that testing in a research setting, but that gets complicated. So there's a couple options. One, you could try to test the tumor and some type of normal tissue that may be available to see if it's a case. It may not be possible it may be logistically impossible. So then could be tested. So this, I think the question is sisters, is that right?
Yeah, 2 sisters.
So the sisters could be tested, particularly if maybe they were older. It's a little hard to test young children, or the parents could be tested. So if both parents are available and both parents could be tested, they do not have a mutation that we could detect, then there isn't a chance the mutation could be passed on to the children. So let's just say these were young girls, or young. I would suggest to test both parents. If the children happen to be, in their 20s or 30s, you could also test the sisters, but the sisters couldn't get the mutation unless it came from one of the parents. And this is the exact thing that genetic counselors are very well suited to discuss. And some disagree with me and say, the other thing, I'm a GYN oncologist, I do a lot of genetics research, but I'm not a genetic counselor. But this is a perfect person who really should meet with a genetic counselor. Most major academic centers have genetics programs nowadays. And again, depending where you live, we could suggest a center that might be worth a few hours drive if you don't live in a major city. But this family should really go talk to a genetic counselor. And we don't know what the odds are. I might guess the odds will be low, but it's an aggressive tumor. So this is the type of family where genetic counseling, I think, makes a lot of sense.
Her question about having their ovaries removed just to, quote on the
side.
Yeah.
I think since we know the gene, we really can test the parents or test them. And if there was not a mutation, I think that would be quite drastic. If there is a mutation, that's a whole other story. And if a mutation was definitely found, what the genetic counselors do sometimes with other people's help is figure out, is it a cancer-causing mutation? Then you might want to see, is it the same mutation that was in the cancer? And if that's really the case, which again is not likely, but certainly worth looking into, if that really was the case, then that's a consideration to be discussed with a GYN oncologist and a cancer geneticist and family, and make a very well thought out decision. But it would be something to consider.
Mm-hmm.
Only mutations identified in the person.
Right. Going to comment about the cell lines for future research. One person said, did molecular profiling through Caris. This woman participated in the TGen study, she said. And would this tool be any use in terms of providing a cell line for future research?
So we only can make cell lines from fresh samples. So once a sample is taken out of someone, usually it gets put in something called formalin, where it's preserved like for pathologic purposes. We can now do sequencing from these samples, which is why me and the TGen group and all the other groups could actually do this study today, which we probably couldn't do 10 years ago. So that would not be useful to make a cell line, but something I didn't think of is if it actually has cancer, if there's any patients who may need to have surgery in the future, or people hear about other people who are scheduled for surgery and are known to have this type of cancer, that's the situation where we could do some coordination to get a piece of the fresh tumor. We can put it in certain media and have it shipped to us to turn into a cell line. And in fact, I was thinking that when most people are initially diagnosed, this is often sort of like expected, of course, but if someone has a recurrence, sometimes surgery is part of that treatment, and that would be a very good time to get some tumor just to study and also to turn into a cell line. So good idea.
Okay. Can you comment on what this mutation may mean for survivors in terms of any relation to any other cancers?
Yeah, like I mentioned, it's related to this malignant pediatric rhabdoid tumor. It will show some features as far as function of these other diseases. And ARID1A is a gene that's mutated in many other cancers. The difference is that ARID1A, for example, in many cancers comes along with a whole host of other mutations, and it's quite complicated to figure out how they interact. With this tumor type and the pediatric rhabdoid tumor, these are only mutations, which is why they're definitely tricky things to target. I hope and kind of think we can do it, but it's possible that we can't. But at least we know exactly where to look, and we don't have to worry that we're going to hit this gene and there's going to be further genes that we're ignoring. As of now, we're pretty confident that this is the main player, and so that'll be very helpful.
Great. I think we'll. Ovie, I want to thank you so much for your presentation. Really wonderful. And I want to thank everybody who called in today for taking the time to do so. And please take a second to fill out the survey that will appear on your screen at the conclusion of the event. Your feedback is always very helpful to us. And again, if any of you on the phone have any follow-up questions or anything, feel free to send them to me. You can find contact info on the OCRF website. And for more information, you can always, our website address is www.ocrf.org. So thank you so much, everybody, and have a great evening.
Thank you, Sarah.
Thanks.
Okay, bye-bye, good night.